Um, Hi everybody. I think we have quite a number of people on the call now, and we would like to start. So my name is Francis Agama, and many of you have received emails from me regarding this bioconductor workshop we are organizing. So first of all, I would like to thank the bioconductor team, the H3 Binary team, and the H3Africa Coordinating Center team for working together to put up this free inaugural virtual bioconductor workshop in Africa. And so on behalf of the team, I would like to welcome all participants and trainers on the call for this workshop. Um, we have about 3 trainers, well experienced, and will be taking us through this workshop. And the aim is that they will help us lay the foundation on how to efficiently use the bioconductor open source resources for bioinformatics analysis. So I would like to give a brief bios for the trainers with no specific order of preference. So I'll start with Professor Son Davis. So Professor Davis received his Bachelor of Science in Mechanical and Aerospace Engineering from the Princeton University and his MD and PhD from the University of Pittsburgh in the School of Medicine and Public Health, respectively. He completed his residency in pediatrics at the Children's Hospital and Regional Medical Center, followed by pediatric hematology or oncology training in the Johns Hopkins School of Medicine National Cancer Institute Joint Fellowship. So our next trainer is Associate Professor Levi Wadron, and he's an Associate Professor of Epidemiology and Biostatistics at the Carney Graduate School of Public Health and Health Policy in New York. He completed a PhD at the University of Toronto and postdoctoral training at the Harvard School of Public Health and Dana-Farber Cancer Institute. His methodological research interest focuses on creating well-documented and user-friendly methods, data structures, and databases to make publicly available genomics and human microbiome data more available and widely used to alleviate the burden of disease. Our third trainer is Associate Professor Benjamin Hyde Keynes, and he is a senior scientist at the Princess Margaret Cancer Center, University Health Network, and associate professor in medical biophysics department of the University of Toronto. So he earned his PhD in bioinformatics at the Université Libre de Belgium, yeah, supported by Fulbright Award. He did his postdoctoral fellowship at the Dana-Farber Cancer Institute and Harvard School of Public Health in the United States of America. His research focuses on the integration of high throughput data from various sources to simultaneously analyze multiple phases of cancer carcinogenesis. Yeah. His team is analyzing large-scale radiological and pharmacogenomic data sets to develop new prognostic and predictive models to improve cancer care. So without much ado, I would like to hand over to Matt Ritchie, and he will take it up from there. Then it will be followed up by the trainers for the workshop. Once again, thank to everybody for signing up and joining us for this workshop. Thank you. Hi, Matt, are you there? Seems to be. Oh, we may have lost Matt on the call. So
Yes, I think we lost them. I think maybe let's give it like 2 minutes and see if it comes back in. There might be an issue with Wi-Fi.
Oh, okay. But then in the meantime, the course materials, there's a link in the chat for all the course materials. So you can access it. And I'll also put it in the Discord platform as well.
So maybe I'll just clarify one thing in the meantime, that that this workshop is actually called Public Data Resources in Bioconductor. The Bioconductor Annotation Resources is another workshop that was presented at BioC 2020 that focused on genomic annotations, annotations of genes, regulatory regions, and things like this. This is a more general introduction to public data resources and different ways of accessing different types of public data in Bioconductor.
Okay, great. So while Matt is not on the call, will you be starting with a workshop, or Sean will be?
So I'll give a few words of introduction, and then I'll hand it over to Sean to start with materials.
Okay.
So welcome everyone, thanks for coming. This is a really new experience for us. Ben, Sean, and I have given this workshop several times before at Bioconductor conferences, but we were always together when we did it. And this time we're all in different time zones. So it's been a bit of a challenge coordinating ourselves. I'm in the Central European time zone, Sean is in the Mountain time zone in the US, and Ben is in the Eastern time zone. And Ben won't be able to join us until after the first hour. So we've arranged our order, rearranged our order a little bit that way, and Sean is going to get us started. But as I mentioned, the goal of this workshop is to introduce a pretty wide range of bioconductor packages for finding, accessing, and using large-scale public data resources. And these include, I think, the most widely used resources in Bioconductor that cover an incredibly broad range of data types and diseases and biological studies. The Gene Expression Omnibus, GEO, is one of the longest running and still, I believe, most used data packages in Bioconductor. It was created by Sean Davis, and he'll give us an overview of that. I believe this package has been around for more than 10 years. I forget how long, but I used it when I first started using Bioconductor. So I love this package. It is still relevant. It started out being about microarray data. There are now count matrices from RNA-seq data, single-cell RNA-seq. There's still all kinds of new and interesting data being posted in the Gene Expression Omnibus. And then the Genomic Data Commons, Sean will also present about this. This is a source for a broader range of multi-omics data. It includes the Cancer Genome Atlas and a number of other NCBI data sets. And then we'll have a range so these are not so much curated resources. These are for direct access to to NIH databases. And then Ben and then I will talk about some Bioconductor hosted curated data resources, ones where there's sort of more more curation, standardization, and reforming of the data for metagenomics, for microbiome studies, pharmacogenomics, and the Cancer Genome Atlas. So there's a few ways you can participate in the workshop. You can view the materials here, although I'll say that my very most recent materials, I believe they have updated. It's taken about 2 hours to do the last update and get it pushed onto this site. But this is built materials of all of the code that will be either running or presenting to you today are here. You can install all of the packages for this workshop on your own computer by doing this. BioC Manager is the package installer for Bioconductor. You use install.packages to install it because it's a CRAN, Comprehensive Archive Network package. And then once that is installed, you use it to install Bioconductor packages as well as GitHub packages. And if you do this command, you will have not only the documentation from this workshop installed on your computer, but all of the dependencies, all of the packages that we use for this workshop will be installed on your own computer. And then finally, you can run the workshop in the cloud. This is an instance of RStudio that runs in the cloud that Sean Davis has created. Not only of this workshop, but actually all of the workshops from BioC 2020 and EuroBioC 2021, and then it will also have all of the workshops from EuroBioC 2020. It will also have all of the workshops from this year's BioC 2021. And this is a teaching environment. These are, you don't have long-lasting disks here, but once you start up a workshop here, you have 8 hours to work on it, or after it has been idle for 8 hours, then that machine and anything that you've done there will disappear. So it's not a place to store your code for keeping for later, but it's a place where the packages are already installed and you can run this workshop without having
Let me, sorry, you're on mute. Sorry, I'll be on mute.
Oh, sorry. There. I was going to say, if you hold on just a little bit on this, then I will be sure. It's been pushed to Docker, so you should be fine. You should have the very most recent workshop materials if you go here now and launch the workshop. Do you want to talk about that later, Sean? Or maybe I'll leave that up to you when to talk about that and hand the mic over to you.
Yeah, go ahead and wait, and I'll go through that.
Okay. And I'll wait to go through these other slides of mine until I'll do those before I present any of my materials. So I'll hand it over to you now.
All right. So it's a pleasure to see all these faces and all these names, and I'll be the first to admit that some of your names will be really challenging for me to pronounce. There are so many people. Normally we try to do introductions here, but there are a lot of people. That's very exciting. I think we're up to 80 some participants now. So I'm not going to go through and do introductions, but feel free to reach out to us via email. The chat here is great, although it's hard to multitask. So for those of you who haven't thought about joining the Bioconductor community sort of more formally, we do have a Slack a Slack channel for Bioconductor. You can find it on the website, how to join, and I'd encourage you to do so. And that'll allow you to follow up with us and also with each other. There's actually a H3Africa channel specifically devoted to discussions around Bioc in Africa. So lots of opportunities here for networking with each other, as well as networking with folks like us who've been in Bioconductor for quite a while. So just giving that communication background. I think what we're going to try to do is we're going to keep you all tied to Zoom for exactly 3 hours. You're not allowed to leave your no. We're going to try to keep this relatively lightweight. So what we'll end up doing is splitting this up roughly into 3 parts. I'll do my part around accessing NCBI genomics resources. Then I'm going to pause and let you folks work on your own, going through the vignette materials. And then after, say, 20 minutes or half an hour, we'll have Levi pick up, and we'll give you times as we go along. Just we'll keep it flexible. But the idea is that there will be some quiet time that you can get up, move around, leave Zoom for a little bit, work on stuff, and then come back. And then I would say the other thing is that these materials are online, and Orchestra, the platform that we're going to be using, is up and running pretty much all the time. And then finally, as Levi pointed out, you can always come back to these materials with your own laptop and work through them at your own pace. There's no way that we can get through everything in our workshop in 3 hours. So there will be plenty of time. There will be some time where we won't necessarily get done with everything, and that's okay. You can come back later. If there are any questions, feel free to drop them in the chat right now. Otherwise, I'm going to show you how to get to the Orchestra app so that you can get to the RStudio instance that we've set up for you. In the chat, a couple of different times is a web link to app.orchestra.cancerdataSci.org. And I'm going to go ahead and share my screen. So when I go to that website, you'll see this kind of login page, or this kind of page. I'm going to log in to get started. Sorry for the welcome to my awesome app. It is an awesome app, but that's not the name of the app, actually. So we're going to sign in. You can sign in with Google, or you can sign in with email. I'm going to go ahead and sign in with Google. If you sign in with email, if this is the first time you're using Orchestra, you'll type your email, and then likely Orchestra will ask you will send you an email to that email address, which then you can use to finish creating the account. But if you have Google, this is the easy way to do it. I have to click sign in with Google here. This is going to pop up now with a list of possible workshops. Any of these workshops are available at any time. There are, I think, 50 or 51 of them now. And a workshop really is just an RStudio instance, which we'll see in a second, and a website basically that describes those workshop materials, and has tutorials to go through. The workshop that we're going to be working on is called public data. So I'm going to do a search in the top right here for public. And we'll see that there's this public data resources in Bioconductor. If I click this link, I'll do that in a second, because I can do that now. This is going to get you to the workshop materials. Hopefully you're seeing my workshop materials right now. And this has the link to the slides, and also has this link to the workshop. So if you want to do this on your own, you can navigate to this site and work. Yep. Mm-hmm.
So can I jump in for 2 seconds? Some people are
yes, please.
Yeah, I wanna find out how many people cannot hear, because I can hear Sean clearly. How many people cannot hear? If you can't hear him, please check your device and increase the volume. But do let me know how many of you cannot hear. Please type in the chat box if you cannot hear. Can you hear me? Because so you can hear. Okay, so you can hear now. Okay. So if you can hear clearly, please log out and log back in. We'll accept you into the room. But a lot of people can hear him because I'm speaking now and you can hear me, right? Okay, thank you. Continue, Sean.
Sorry about that. I'll also try to speak up. I sometimes end up muttering a little bit, and it is 6 in the morning here, so I haven't had my full cup of coffee yet. So the workshop materials are available. You can come back to them anytime you like, and there's a lot of material in here that can be useful for understanding not just how to use the packages, but why the packages were created, some of the analytical capabilities, etc. I'm going to jump back over here to choose workshop. And now if I click launch, what this is going to get me is down in the center here. This is going to get me my own personal version of public data resources in Bioconductor. Once the bar stops going by, you'll see a launch workshop pop up. This may take anywhere from a second or two to maybe a minute, depending on how much work Google has to do to get resources available. So I'm going to launch the workshop now. Notice the username and password are the same, super secret for all of us. RStudio. All right, and so we've landed in RStudio. There's nothing different about this RStudio, my RStudio that I'm seeing, and the RStudio that you're seeing. And there's one special thing about the way that we create these environments. All the files that we need for files and software that we need installed for running this workshop are already available to us. So to cut down on typing a little bit, we're going to jump over here to vignettes. And this public data resources.rmd is the actual workshop that we're going to be doing. And we'll see it now loaded over here. I'm going to pause for a second here to see if folks are kind of caught up. I've got a small screen today, so let me know. Let me see. Get the chat here. So let me know if you need me to slow down. Levi, if you're looking at chat, if you can actually, I think I can open it now.
Yeah, I'm keeping an eye out.
Cool. Okay. All right, cool. I'm going to go ahead then and start walking through this a little bit. I'm going to start with assuming that folks have not used RStudio much before, and just quickly walk through what RStudio has to offer. So RStudio itself, which is what's in front of us, is not R. It's different. But what it does is gives us access to R running in a separate process. So R is running in the background here. It's actually running in the cloud in this case. But the only place that we're going to actually interact directly with R is in this bottom left corner in the console. So if I type help, for example, what that's done is to tell R to open help. And in this help panel in the bottom right-hand side, I'm seeing the help for help. So anytime that you have questions about what we're doing or about a function or something that we're typing, you can always do this is help on print. And that'll pop up over here. The other way to get help is a question mark followed by the thing that we want to get help on. So when you see things that don't make sense, or you see a function that you haven't seen before, it's a good idea to try help. We can also, you've already seen the file browser. Any plots that we create will potentially show up here. Installed packages are available under the Packages tab. We can also install additional packages through here. RStudio also has a little viewer tab. This viewer is, you can think of as sort of a mini web page. So if there are things that R is going to generate that are best viewed on the web, you'll often see them pop up in the viewer. I'm not going to spend much time talking about what's in the upper right-hand corner here, other than to note that as we go through things, R is going to be collecting a history of all the things that we've typed. At the end of this session, you might want to, for example, save history to a file. I'm going to go ahead and do that now. And now if I go back over to files here, I think that saved it in my home directory. So there should be a myhistory.txt. One thing that I can do here when I'm done is to I'm forgetting here how to download these things. We'll come back to that. All right. The last thing that we're going to talk about is the important part, which is in the upper left-hand corner, this public data resources.rmarkdown, .rmd. So this is the actual workshop materials that you see on the other website. So if we take this and run it through R, then we're going to get this. Actually, we're going to get, yes, we'll get this, the workshop materials. So this and this are just two different formats for the same material, with one exception. the material on the Public Data Resources website has been run through R, and all the R code's actually been run. But what we're going to do here is instead step through the workshop materials a piece at a time, and this will allow you guys to evaluate the code, play with it, and go off on tangents, substitute new variable names, experiment basically. We've already installed all of this software, so we're going to skip over this part about installation. And the first thing we're going to talk about is Geoquery. Geoquery accesses the NCBI gene expression omnibus. The gene expression omnibus is very large at this point. I think it's around 4 million samples of gene expression or ChIP-seq data. Anything that's what we might call functional genomics is likely to land in NCBI Geo. And if I'm not going to go into much detail about how NCBI Geo is set up, but suffice it to say that Geo uses its own proprietary formats for storing data, and all Geoquery, the software that we wrote back in 2007, all Geoquery does is to take an accession, an ID from the NCBI gene expression omnibus, and load those data directly into Bioconductor. And the idea is to make the data in Geo as accessible, valuable, and reusable as possible with as little work as possible. So Geo is laid out in really as 3 separate types of data. There's the simplest one to think about is the sample. So the sample is essentially all the measurements that come from either a microarray or potentially from RNA-seq, etc. The platform is the description of the genes that are, or the genes or chromosome locations, etc., that are available for measurement for that sample. So a sample is related to a platform. And then to join samples together into an experiment or a set of samples, there's this concept of a series. So a series contains samples, and a sample has a platform. Most of this is not very useful or valuable to us because Geoquery takes care of all of the details of dealing with these relationships. You can read through here what the details of samples, platforms, series, data sets are, but we're going to jump right to it here, getting started using Geoquery. Now, the way I'm going to do this is I'm going to walk through quickly the starting pieces of material. I want you to just watch, and then I'm going to pause for about 20 minutes or half an hour and let you work through this stuff. I'll watch the chat and watch Slack. And if you have questions along the way, we can either do the questions online or through the chat. But the idea is that I'm going to be quiet and let you work. So to get started, let me show you how to use R Markdown, this R Markdown document to do work. If I go to a line like this grayed out box with the 3 ticks around it, this is actually R code. So if I type Control Enter, so Control and then Enter or Control Return, depending which keyboard looks like, what this is going to do is to take that line, this line, and put it down into the console down here. So by hitting Control Enter, it's essentially the same as cut and paste, just a little bit quicker. So what we're going to be doing in this Geoquery session is, thanks, Levi. Levi's pointing to how to get to the materials: vignettes, public data resources, and you'll have this public data resources file open in the upper left-hand corner. What we're going to do is to make a multidimensional scaling plot of cancer data. You can go to this web link and get information about the actual study. But at this point, I'll stop discussing this and just say that what you're going to do now is to walk through the code here by hitting Control Enter again. Geoquery is going to do its thing, and as you're walking through, you can read the description of what you're doing. The goal is to produce the multidimensional scaling plot that's available here. So by going through this material, this is what you're going to end up with. And of course, this is just an example. If you have a data set that you're interested in, after searching the Geo website or finding a paper that's interesting that has available data, you can, adapt this analysis to your own work. I'm going to stay on this web page now that I've shown you how to interact with RStudio. And the next thing we're going to work through is the Genomic Data Commons. So Levi is going to show you a much easier way of dealing with data from cancer data from NCI than what this package is going to show. But for those of you who spend a lot of time working on cancer data, it might be useful to have the ability to really dig into all the cancer data available at the Genomic Data Commons. So what is the Genomic Data Commons? You can think of the Genomic Data Commons as kind of like the NCBI Geo, but for the National Cancer Institute's genomic data only. So this is very specific to cancer, very specific to NCI, the National Cancer Institute. And unlike Geoquery, it's not, or unlike NCBI Geo, it's not a generic data resource. It is specific to cancer, and it's much more complex in the sense that as we scroll down to the data model that the GDC uses to store the data and the metadata, you can see that the data model is pretty complex. For example, there's not just a sample, a platform, and a series. There's a sample, there's a case, there's a clinical supplement, there's a tissue source site. All of these things are things that you can look up and use to access the data. And what the Genomic Data Commons package does is to try to make it relatively straightforward to query this data model to get the kind of information out of the GDC that you want. For those of you who don't spend a lot of time dealing deeply with cancer data, feel free to go through this, but I'll be honest, this package is really meant more for people who are developing software that use cancer data than actual people who are trying to use cancer data directly. The one thing for those of you who might be sort of more software developer kinds of folks, this is a kind of cool package because it accesses an online web-based API. So a lot of the things that you'll be typing or cutting and pasting in RStudio are actually not doing much work in R. They're calling out to website, basically to a web API that does most of the work. So what this is going to show you, what this section is going to do is to show you how you can use the Genomic Data Commons package to learn things about what's available in the NCI Genomic Data Commons repository. I'm going to stop there. Let me see if I can see any obvious questions.
So far, I've been able to keep pretty much up with them in the chat.
All right, thanks. Okay, then I think what I'm going to do, it's 48 minutes after the hour. Why don't we work until about 15 minutes after the next hour? So about, actually, let's make it 10 minutes after the next hour. So we'll be working for the next 22 minutes. And what I really want you to do is to go through the vignette, cutting and pasting. But more importantly, if there are things that you want to try, experiment with, this is a great time to do it. So don't feel like you need to go through all the materials. Go through the parts of materials that you think are going to be relevant to you, and spend more time on the stuff that looks interesting. And then we'll pick up again with Levi discussing the simpler and faster way of accessing curated cancer data, among other things, at around 10 minutes after the hour.
Actually, probably I'll put Ben next, since he's here and has to leave again later, and then I'll finish.
All right, so around 10 minutes after the hour, somebody will come up next.
And we'll stay here monitoring everything.
Yep.
So you can raise your hand if you want to ask a question, or just type it into the text box or the chat.
Okay.
I'm going to make a quick comment. I told you I'd keep quiet, but you'll notice on my screen in the bottom left, there are 102 parsing failures. This is actually not that uncommon with data coming from Geo, that a few of the lines look different than others. I've designed Geoquery to be fairly robust to this kind of thing. So in general, these are going to be warnings that you can ignore. So despite the fact that it says parsing failures, you can go ahead and get or go ahead and proceed, the data will have loaded correctly. Those parsing failures, like I said, it's just a function of the way that Geo uses rows. They sometimes are a little different as you go down through the file. Kevin, if you glance up, yeah, exactly. If you glance up at my screen, or glance at the screen, you can see what Levi's pointing at. So if I click on, if I scroll down to one of these gray areas, this is R code. So I can do one of three things. I can click here and click run over here. I can click run here, or I can hit control and return at the same time. And you'll see that that line got translated or got typed, essentially cut and paste into the console.
Sean, how much RAM do these Orchestra instances have?
Just to give me a second and I'll show you guys what's going on.
That's a pretty cool screen you've got there, Sean.
So this is what Orchestra is doing in the background. And Levi, you were asking cluster about memory. So we have 100 gig currently used by everyone here in total, and we have 500 gig of RAM available to us right now. We're actively using 17 cores out of the 128 that we have allocated, and we have almost 6 terabytes of available disk, and we're using 400 gigabytes of that right now.
And that's what about 80 people? So each person is only using a little over a gigabyte of RAM?
Correct. Yeah.
So that was one of the questions before, how much RAM they would need to run this on their own computers. So 8 should be enough. It doesn't use a lot of RAM.
It does not use a lot of RAM. Yeah.
Yes, there is one, I believe there was one bug fix that I made that didn't quite make it into Orchestra before this started, which is a library Magritte, which is being used in there. So, but I believe it's installed anyways if you do library Magritte.
For those of you who might be joining us a little bit late, you can watch my screen for a second. I'll show you what's next after launching RStudio. So in RStudio, after you get started, or after you launch it, in the bottom right, you'll see files. I'm going to click on vignettes. And inside the vignettes folder, we're going to see this public data resources.rmd markdown file. If I click on that, in this upper left-hand corner, I'll have that file show up. And this file is the same, is what ends up being the website workshop materials. To use this file to walk through the material, you can scroll down to one of these gray areas. This is our code inside these tick marks. And if you click on one of these lines, you can do one of three things. You can click the green button, which will run the lines inside this gray area. You can click run current chunk, or you can hit control and return. And that will copy this line down to the console. You just keep walking through, reading as you go along. You can keep hitting control enter as you go along, and that'll keep running our code.
BD.
Before we bring Ben up to the stage, I just want to say how exciting this is to be offering a workshop to people in Africa. It's to me a wonder of modern technology and something that I wouldn't have even thought about a couple of years ago. So I just want to thank the organizers and the attendees for making this possible. It's really exciting.
And I'll just add to that. You know, we at Bioconductor don't own this stuff. So for those of you who have who are attending and have other folks at your university or hospital or wherever you're working, have folks who you think would benefit from this kind of thing, feel free to offer these courses yourself. It's really just a question of finding a Zoom room, and if you have trouble doing that, let us know. And these workshop materials are always available. So to the extent that you want to reuse these for your own purposes and teach amongst yourselves, we really would love to see that happen. So don't feel like you need to wait for us to organize these kinds of things.
Yeah, so it's exciting to hear that actually we can use the materials for this workshop, and yeah, so we will certainly extend the materials to others who are not able to join. Because I see there are a lot of like design workshops on the Orchestra platform. Yeah, but then the question is, for example, after this workshop, and can the participants for this workshop be able to continue to access the Orchestra platform?
Yes, we have funding from both NIH and NCI to make these workshops available through Orchestra. With the 8-hour time limit, and that tends to vary a little bit. Sometimes I'll change it to 4. The workshops are always available, and people can view them, use them as often as they like.
Great. Exciting to know that. Yeah, and also thank you to you guys for making yourself available to organize this workshop, because I'm very much aware of the difference in time zones, and even trying to get everybody for like a meeting was a little bit challenging. But then here we are, yeah. So thanks to you guys too.
Mm-hmm. Yeah, it's an honor.
It really is. Mm-hmm.
There's a bunch of errors coming up from not having libraries loaded, and that's because we didn't actually load all of them at the start. There's a chunk at the top that installs the packages locally, which you don't need to do on Orchestra, but you do need to load them all using library. and one that was missing in the version that many of you are running is, yes, library Magritte. It's, by the time I pulled Orchestra, it was already there, but it was a little bit late arriving. The next time you start this up, the library Magritte will be there.
Yes, I see a question from Polycarp. Will you attend to that?
It's around 7:15, so I think we're going to go ahead and turn things over slowly to Ben. I think he's ready, and feel free to continue to work through these materials, the Geoquery and Genomic Data Commons materials, or to jump ahead to Levi's materials, or to spend all your time on pharmacogenomics data if you like. So don't feel like there's a teacher watching you. You do what works for you. So.
Do you happen to know, Sean, where the plots appear here? I haven't actually gotten that far on Orchestra to know. Should they appear in the plots panel, in the code panel, or I guess it would either be the plots panel or in the code panel, depending on what the default settings.
I'm going to walk through the rest. I'll walk through this really quickly. It's actually pretty quick to run this. And then finally, the plot itself. And it will end up in the if you're doing what I'm doing, which is to evaluate the code in the R Markdown document, it'll end up in the R Markdown the R Markdown window, right in line with the code. So here's what, if you're still looking at my screen, here's where it ends up, right underneath the code. If you want to see it bigger, there's a little button here, show a new window. You're not going to see that, but it'll pop up in your window. And will be a larger version of the same thing. All right. I'll hand off to Ben at this point, and we'll go from there.
Thank you, Sean. Can you hear me?
Yes.
Perfect. Thanks. A great introduction, by the way. Introduction to our studio and the Orchestra platform. So I'll share my screen. I'll go through a few slides. I'll try to go slow, so don't hesitate to interrupt me and ask really any questions. There are no stupid questions, only stupid answer. So I'll be the one to blame. Okay, so today we'll discuss, this part of the workshop is about pharmacogenomics, and I'll explain what pharmacogenomics is, but hopefully some of you already dealt with this kind of data. So pharmacogenomics is a field where we study the effect of drug treatment on organism or disease models such as cancer. And so it's usually used to discover biomarkers. So those are any kind of molecular entity you can measure and that you can use to predict whether a specific type of cell will respond to a drug, whether depending on the phenotype. In cancer, we try to kill those cells. So we try to find drugs that actually inhibit the growth of the cancer. And so the problem with pharmacogenomics is that it's multimodal. It has multiple data types. It requires multiple data types to really do it right. So not only a lot of molecular data, such as gene expression, mutation, copy number variation for a given, let's say, cancer cell. I'll focus on cancer today. But you also need the rather complex pharmacological profile. So we treat those cells with drugs and we measure viability. And so you have all those data types that need to be in sync somehow for you to do your statistical or machine learning analysis. And in the field, we have a lot of standards when it comes to the molecular data. We know very well how to annotate the genome at the loci region or at the gene level, whether it's mutation, copy number variation, or gene expression, for instance. When it comes to drugs, on the other hand, it's much more complicated. There are different representations of the drug structure. The drug name is not always standard. And it's somewhat also a bit of an issue for the cell lines themselves. It always amazed me that after decades, I think it's over 60 years now that the HeLa cell line has been created. After 60 years of research on those cell lines, we still don't really fully agree on how to name them. And so it becomes a little bit of an issue when you start analyzing those data. The same cell lines may have different names or different annotations. So we decided to basically implement a R package to solve to address some of those issues. And this package is called Pharmacogex. And we aim it, it's completely open source, it's on Bioconductor, and we aim it as an easy to use package to access highly curated data, but also analyze those data to some extent. And I'll show you how you can download a data set. And the beauty with those data sets is that we spend years within my lab, as well as with the help of the community, to create those data. So hopefully it is pretty clean. We've used standards to annotate the cell lines and the drugs, so you don't really have to take care of that anymore. You can really just focus on the analysis. And when we started to work on Pharmacogex, it's actually triggered a whole set of research activities in the lab that are very, very closely related. So Pharmacogex is really focusing on immortalized cancer cell lines screened with drugs. But then we realized that a lot of drugs are also being screened in vivo models, such as patient-derived xenograft or genetically engineered mouse models. And so those are very different kinds of data. Usually we measure volume over time in those in vivo models. So it required another set of functions and another way to store the data. So we developed a package called Xiva for those in vivo pharmacogenomics data. Then we also realized that people are actually now doing high throughput radiation screening on those cell lines. So they irradiate those cell lines and see what happens. And some cell lines are very resistant to radiation. Some cell lines are not. So we, again, a slightly different way, experimental design and way to perturb the cell lines. So we created the package called RadioGX. And then the last package of the series is called Toxico Gx. It's very similar to Pharmacogex, except that it's really focusing on toxicogenomics. So the study of the side effects, the consequences of a chemical exposure. So the goal here is not to kill cancer cell lines. It's actually almost the opposite. Is we study kidney and liver cells, and we hope to see actually no effect upon chemical exposure. So it's kind of the antithesis of pharmacogenomics in a way, but we also realized that there were a lot of commonalities, and the field was also struggling with the same issues of standardization, hard to access data, and the need for a high level of curations. So we'll focus on Pharmacogex today, but if you find it interesting, you can also investigate the Xiva, Radio GX, and Toxico Gx packages that are all on Bioconductor as well, and they're all open source. So any questions so far? So I don't see the chat window, but don't hesitate to just speak up. And Sean and Levi, if you see a question, just interrupt me and I can answer it right away.
Yep, will do.
Gotcha.
Okay. Okay, so just to give you a little bit of an overview of how rich the field of pharmacogenomics is, what really kind of get us interested, my lab is focusing more on the bioinformatics and machine learning analysis. We're no biologists, we're no pharmacologists, but we really kind of specialize on analyzing those high dimensional complex multimodal data. And so when we saw all those cell line-based drug screening data being released in the public domain, we got really excited. You can see here, it all started in the late 90s with the NCI-60 Developmental Therapeutics Program, one of the biggest drug screening program in the world, where they took a small panel of 60 cell lines and they started to screen a lot of drugs. They actually have more than 200,000 in the database, but there are 60,000 drugs that have been screened on almost all the complete panel of 59/60 cell lines. So it's a gigantic data set. Even so, the set of cell lines is relatively limited. It's a lot of drugs that they've screened. So it's a very rich source of data. And obviously, they've profiled those cell lines at the molecular level, I would say, to exhaustion. They have basically virtually all the data types you can dream of: gene expression, exome sequencing, whole genome sequencing, I believe, methylation, metabolomics, probably. They have a very, very rich set of molecular data you can mine. The Japanese actually did the same in the late 90s with their own set of 39 cell lines, a much smaller set of drugs. And you can see that over the years, more data sets have been released by the public to the public domain. And what's very interesting is that it's not just academic research institutions, but it's also companies. You can see here, GlaxoSmithKline, Pfizer, Global Pharmaceutics, Novartis helped the Broad to release data, Genentech with the GCSI dataset, AstraZeneca working with the Sanger. So it's really this kind of rich ecosystem where both industry and academia contribute data to the scientific community. And you can see here the number of cell lines and drugs being screened and included in those data sets is really increasing rapidly. And if you take the sum of those data sets, you have a lot of data to play with, which usually means a lot of troubles to, a lot of issues to address. But hopefully Pharmacogex will alleviate some of those issues for you.
Ben, there was a question.
Yep.
I guess it came from your discussion of, you mentioned LD50, but someone asked if Pharmacogex can be used to predict the drug half-life. So maybe just clarify what you were talking about there.
Oh, that's a very good question. No, it doesn't. So we don't focus on pharmacokinetics. We focus on pharmacodynamics. So we really look at the effect of the drug on the system, but it's not really used for PK studies, unfortunately. So I'm not sure that's really something we want to do in the future. There might be other software packages doing something like this. We honestly don't have the data. It's a completely kind of separate set of data sets. So here it's really, the input is kind of the molecular characteristics of the samples you're screening, you're treating with the drug, and the effect, the phenotype. And to be honest, the phenotype is fairly limited for now. We only focus on cell viability, usually assessed via measurements of the ATP level in the plate or the DNA level as a surrogate for whether cells are viable or not. So there's so much more to do there. You can look at pharmacokinetics, you can look at very different phenotypes such as migration, mobility, size of the cells. And there are data sets being released now using high content microscopy that we plan to include in the future. and the complexity of the data will really kind of increase exponentially from now. I think we have much better tools to look at the effect of drugs that we had in the last 20 years. So it's an exciting field, but Pharmacogex is really focusing on this molecular plus cell viability measurements. Any other questions? No. I'll move on then. Okay, so as I said, Pharmacogex is actively developed in the lab and maintained, and actually a very talented software developer, Christopher Eels, you see here in the author, is really single-handedly improving Pharmacogex and supported by Peter Smirnov from a scientific perspective. So we are modifying the package quite a bit, and I'm very excited to say that the next version will be able to handle drug combinations, which is also a set of data that is being released quite actively nowadays. And we are going to replace our list of summarized experiments for the molecular data with the great multi-assay experiments package developed by Levi's group. So stay tuned, but for the moment, you can already access and deal with quite a bit of data with the current version of Pharmacogex. So as I said, Pharmacogenomics is really combining two sets of data: the molecular data and the drug response, the cell viability data. By drug response here, I mean this beautiful drug dose response curves. So on the x-axis, the drug is being tested at different concentration. So you increase the concentration of the drug and you see what the effect is. On the y-axis, you have the actual effect that is being measured, so the percentage of cell viability. So if you have no concentration of the drug, so that's the control, the cancer cell lines is happily growing. So that should start at 100% viability if you normalize it properly. And then you increase the drug concentration, and what you see is an effect on the system, where if in that case, that drug seems to have an effect where the viability decreases with respect to the drug concentration. That is a rather complex phenotype. You have to model that curve. There are many edge cases. If the cell line is completely resistant, you have a flat line and 100%. So we usually summarize those data with a single number, and I'll probably showcase one of those metrics in the next slide. But we summarize that curve in a single number that reflects how responsive the cell lines is for that drug. And we basically correlate this phenotype to the molecular profiles to find biomarkers. And so we've tried our best to create as many data sets as possible. You see there is a very long list of data sets. Resources allowing, we created a fair bit of those, the biggest ones. And we also uploaded all of them in Zenodo, actually. And I would like to show briefly our Orchestra platform. So if you can still see my screen, I'll quickly go on Orchestra. So not to be confused with the Orchestra with the H platform that you're using for RStudio. Unfortunately, we use the same acronym without the H. And that's our platform to orchestrate all the data creation activities. So if you click on the first icon, the pharmacogenomics data, you can actually you have a bit of a dashboard to see how many data sets we've curated, how many pipelines we use to analyze the data. If you click on the canonical psets on orchestra.ca, you actually have conveniently a set of psets that we use in the lab. So we consider those ones like the best data set for our research. So if you click on CCLE, for instance, the Cancer Cell Line Encyclopedia, you have a bit of summary here. And this is also available as a Zenodo. Here's the Zenodo digital identifier. I guess if we click here, we should end up on Zenodo. And what's special? Yeah, here we go. It's a fairly big data set, so downloading it will be a bit of a hassle today, but you can do it in your own session. What's great about Orchestra is that in the background, we keep all the scripts and the data provenance information to make sure that we can always reproduce that exact data set. So it's kind of a fancy, sophisticated way to curate your data set in a fully automated manner. So if you make a mistake, you can always go back. The opposite of that would be to open the data in Excel and modify them and save that Excel file. That's a recipe for catastrophe. I can tell you, your gene name is going to be modified probably, and you won't be able to really trace all the modifications that you've done to the data. So say that again. Sorry, please ask your question again. I didn't hear it.
I think it might have been.
I think that might have just been noise. Yeah.
Oh, okay. Okay. So from here, you can easily. This is all the data sets you can have access to with Pharmacogex. They will have a slightly different names now because we keep improving them. So the available pset will give you that list. If you take the dataset name and you use the download pset function, you can actually download the data directly to your session. Okay, and so we have two types of data in Pharmacogex. I somewhat maybe misled you a little bit, saying that we only focus on cell viability data. There are two kinds of data set we use. The first one is drug sensitivity data. That's exactly what I described already. So you have the cell lines, you treat with drug, you have the drug dose response curves. And from there, you can look at association between molecular data and drug response. So whether genes are over, if the gene is expressed, does it mean that the cell line will die when treated with a given drug? Maybe if a gene is not expressed, it means that the cell lines will resist to the drug. So you have all kind of association. You can look at it that way. And then we also have included the connectivity map and the L1000 dataset. So those are two big datasets from the Broad Institute where they didn't care much about the effect of the drug on viability. What they've done is to look at the gene expression before and after treatment. And they usually have very kind of low concentration of the drug so that they don't kill the cells, but they want to see how the drug is actually affecting the gene expression profile of those cell lines. And so we call them drug perturbation signatures. It's how the drug is pushing the cell line from a given state to another state. And that could be very informative. And you also have this kind of data in the Toxico Gx package, by the way, where you have kidney and liver cells before and after drug exposure to chemicals. So very, very similar concept. So you have those two kinds of data, but today we'll focus on the drug sensitivity data. And so because those data are multimodal, we basically had to define a new class of object that's basically building on other classes in Bioconductor, as it probably always should be the case. So it's nothing really fancy, but it provides some convenient functionality. So that's what we call a Pharmacoset, or pset in short. So that's a new S4 class that basically contain not only all the molecular profiles, but also all the pharmacological profiles, the pharmacological data, the drug, the cell line, and the cell line annotations. And once you have this pset, which is, again, the results of the creation of those data set, you can do stuff that are you can easily do stuff that would require a lot of curation and time before Pharmacogex. So you could fit the curves, those drug dose response curves. You can visualize, you can fit them. You can actually compute different statistics. So I actually didn't have a slide about that, but IC50, for instance, is the concentration that you need to kill 50% of the cells. And so that's a very commonly used matrix in biomarker discovery, because it kind of summarizes how sensitive the cell is to a given drug. If the IC50 is very high, it means you need very, very high concentration of the drug to kill the cells. if the IC50 is very low, this cell is exquisitely sensitive to a given drug. So the range of IC50 can really discriminate how much drugs you need to kill the cells. The AAC is the area above the curve, above the drug dose response curve. So that's another metric, and that could be used for biomarker discovery. And there are many others that you can compute in Pharmacogex. You can easily extract all the molecular data. For the moment, they're stored in summarized experiments that have been processed using specific pipelines such as Callisto for gene expression, for instance. But you can easily extract those processed data as summarized experiment objects. And then basically now you can easily create this kind of drug phenotype, those molecular phenotype, whether it's mutation, copy number, variation of gene expression, for instance, and then you can do your statistical analysis. We provide a function to do like very simple univariate analysis. So it's basically a linear model looking for the association between drug response, such as IC50 or AAC, and a given molecular data type, such as gene expression, controlled by tissue types, if you have multiple tissue types in your portfolio of cell lines. So cell lines could come from lung cancer, from lung, from the breast, etc. And that could also be associated with drug response. So we actually adjust the model for that. And you can basically develop all your own analysis once you get access to those data. Then it becomes, then you start playing in the wonderful world of machine learning and statistical analysis. And we have no pretension to provide all the best pipelines for you guys. It's still a very active field of research, but we hope to facilitate your work so you can focus on the development of those sophisticated machine learning pipelines, for instance. Okay, so Radio Gx, it's exactly the same thing, but except that instead of a drug dose response curve, we look at the effect of radiation. Usually you have the survival fraction in the y-axis and you have the dose in gray for the radiation. So very, very similar. For the moment, there is only one big data set in that field from the Cleveland Clinic. It was a collaboration with the Broad, but we hope to add more data sets in the future. There are a lot of smaller data sets, but we focus on that big one. And then finally, you have GIVA, which is the equivalent of Pharmacogex, but for in vivo studies, where usually you take a piece of tumor. If it's about patient-derived xenograft, you put it in a mouse, you let the tumor grow, then you split that tumor in multiple mouse, so you have a second generation, and so on and so forth. So at the end of the day, you have enough mice for the control and the drug treatment arm. And you basically let the tumor grow with and without drug treatment and see what happens. So here's an example of tumor growth curve. So in the x-axis, you have time. On the y-axis, you have the volume of the tumor, usually measured as either like pitching using a special measure. You just pitch the tumor to see what the diameter is in multiple directions. So not very precise. If you're part of an institution that has the capacity, you can even do a CT scan and compute the volume of the tumor that way. It's much more precise, but it's much, much more tedious and expensive. And here we've created a few data sets, mainly the PDX encyclopedia, but we are in the process of adding more data sets. So the PDX encyclopedia was released by Novartis. So I think that's probably it. Yeah, that's it for the presentation itself. So do you have any questions regarding the science, the context for Pharmacogex? Okay, if I don't see any questions, maybe we can switch to the R session. I have to apologize, going to be fairly limited today in terms of hands-on experience. I just figured out that all the code was gone from the RMD 5 for the public data resource markdown. So apologies for that, but I'll try to show you a few basic comments so that you can you can see how to download and play a little bit with those data sets. So let me share my screen again. Okay. So do you see the RStudio window?
Yep, we got it.
Okay, perfect. So
if you can make your font just a little bit bigger, that might be useful.
Yes.
Just, perfect.
Okay,
thanks.
Perfect. Okay, so let's start from the beginning. It's going to be a bit of a live session, so forgive me if I take some time to remember some of the functions. But let's start simple. Let's just load the library. It should be preloaded, so it should be no issue. But let us know in the chat window if you face any challenges to load the Pharmacogex library. So part of that dataset, we've actually included two small kind of shrinked version of dataset from the Cancer Cell Line Encyclopedia, for instance. So if you write that command, data CCLE small, you will actually have access to a small pset, and that's part of the package. So it should be very fast to load. If you actually print that object, then you'll get some information about
Ben,
Yep.
May I jump in? There's like two questions in the chat.
Okay.
From Bandi saying, "Hi Ben, you mentioned that you will indicate how to summarize the dose response curve into a single value. Is that coming later?" And Kevin says, "Please, where do we get the documentation for the Pharmaco?" Can you please answer those two questions, Ben? Thank you.
Okay, yeah, I'll try to address them. It's really a pity I don't see the chat window when I'm showing my screen. Okay, first question. Yes, I'll try to show you how to summarize the drug dose response curves, and I'll certainly show you how to directly access the IC50 and AAC values, because we actually pre-computed them in the object. And let's see how do we access the help for the package. So obviously you can look at the vignette online on the Bioconductor website. That's probably very convenient. If you want to see the information about the package itself, you can use this command library help Pharmacogex. So you see the version, the maintainer and the like, and a series of functions. So what I would do if I were you, I usually go online, grab the PDF, the reference manual, the PDF from the Bioconductor website. So I have an exhaustive list of all the functions and the documentations, but they're all here if you want to see them. So for instance, here we have all kinds of functions to compute the IC50, compute the area under or above the curve, et cetera, et cetera. So I'll show you a little bit. And so because this package is now using S4 classes, it's sometimes a little bit tricky to find the documentation for specific functions. So what I like to use is the double question mark to make sure that I do an exhaustive search for the right function. So let me see if I can understand. Oh, that's a little bit slow. Here we go. Okay, so that one was no trouble. The download pset function, you can look at all the documentation directly in RStudio using the question mark function. But some function, I'll show you, some functions are a bit more problematic. For instance, let's see, summarize. Okay, that's a good example. So if I just look at the summarize molecular profiles function, it's going to be fairly disappointing. You're just going to have this kind of wrapper saying, hey, you have to provide an object. And the reason is that we actually have abstracted a lot of the functions in a dependencies, another package called Core Gx. And Core Gx is basically feeding Radio Gx, Toxico Gx, and Pharmacogex with a lot of common functions. So basically, if you really want to have the documentation, you have to basically say, hey, I want the documentation of this function for Pharmacogex using the Pharmacoset methods. And then you get access to the real, full set of documentations. I don't find that very intuitive. So what I do is I use the double question mark help function, and that will give me a set of options whether I want the summarized molecular profiles from Core Gx or for Pharmacogex. So just click here, and I finally get the documentation I want. We are trying to simplify that process a little bit. So we apologize if you find a little bit difficult to find the right documentation, but it was kind of the price to pay for the modularity of the code across our suite of objects. So let's focus on very basic stuff. First, you may want to know what are all the available p sets in Pharmacogex. So if you have no clue, you can use the available p set function, and that will basically return you a rather big table with all the dataset we've created so far. So if you look at this table, you have the dataset name. That's kind of a canonical name we use for plotting and the like. But you have more detailed information about the data being created. So you can see here, it has been created at that time. This is the official Pharmacoset name, and that's the one you should use when you want to download the dataset. I won't download any of those because they usually is gigabytes of several hundreds of megabytes each. So we're going to use the smaller object that I presented before. But you can see here that, for instance, GDSC, the Genomic of Drug Sensitivity in Cancer, it's a data set from the Sanger. You can see that there are multiple versions already. There is one from 2019, version 2.8, version 1.8 from 2019 as well. And then you have a version from 2020, 2.82, 1.82. And so that's a very important aspect of Pharmacogex. We try to keep track of we version all the different versions of the data sets, because those data sets evolve rapidly over time. The Sanger and the Broad, for instance, they tend to add cell lines and add drugs, and we move bad data when they spot some errors in the data. So Orchestra is supposed to help us to really track all the versions of the data and make sure we always know which data set we're dealing with. So you have the official p set name, then you have the version, and you have the type, whether it contains just the sensitivity, the drug dose response curves, whether it contains the perturbation, like the connectivity map. Actually, here you go, connectivity map. It only contains the perturbation. And some data set, interestingly, they have a bit of both. They have data about drug dose response curves, but they also have the G expression before and after. So then you can see both. It's both sensitivity and perturbation. We give you the kind of the seminal publication for each of those data sets. So if you want to write a paper, you know what paper to refer to cite. And we also give you some indication about where we got the data. And more importantly, you get the DOI in Zenodo. So if you want to cite this data set in the paper, always cite the initial paper and acknowledge the source of the data, whether it's the Broad, the Sanger, or a company. But you can also use that digital object identifier to say that's exactly the version of data set I'm using. Okay, so if you wanted to download a data set, you would just go, like this, where you put this official p set name. Where is it? Like, for instance, CCLE 2015. So, okay, I want this data set, and instead of available p set, you would say, hey, I want the CCLE data set using the download p set. So I won't run it because this data set is, I think, over 2 gigabytes. But in basically 2 comments, you know what is available in Pharmacogex, and you can download the p-sets directly as objects. So now, for instance, let's just say, hey, I've downloaded the CCLE dataset, and now I can see what's in there. And in CCLE, actually, you have 1,061 cell lines. You have data about the gene expression of those cell lines. We only put 50 genes for this small object. And you have it for both microarray and RNA-seq. You also have copy number variation. And you should have, in the real data set, you also have mutations. So if you download the official p-set, you'll have much more data. There is no drug perturbation here. But if you look at drug sensitivity, there are 11,670 drug dose response curves. So it's a fairly large data set. So now I'll show you a little bit how to access some of those data. So you may wonder, what kind of cell lines is there in this data set? So we have some convenience functions such as cell info. Where
would you show the downloading step again, or just put it up on your top one while you're working? There was someone who missed it.
Oh, sorry. So if you want to download a data set, you can just use the download pset function, and you have to provide an official data set name. How do I access that? Can I access the chat window while I'm presenting? Do you know, Levi, Sean?
Good question.
I don't think so.
You can't, because you're sharing your screen, you won't be able to see it.
Yeah, no, it doesn't look like I can.
Because you're gonna have to stop sharing, and then therefore you're not gonna be. So we're not gonna see your screen, but you'll be able to see the chat. So you have to choose which one it is.
So that's not very convenient. So again, you were supposed to get all that code in the RMD files. So I didn't really have to cut and paste. So trust me, I'll put all the code in the RMD file, and Levi will share that back with you, so that you always have access to all those comments. But for the moment, I just have to show it live, and I'll share everything with you afterward. Is that okay? So yeah, so if you want to download a pset, just use the download pset function and a string specifying exactly the official name of the dataset, which you find from the available pset function in the column pset name here.
Ben, if you want to get the chat window, if you scroll up to the top of your sharing, you should see a little menu. So now you can click chat and it'll come out floating to the right or floating someplace.
Thanks so much, Sean. I will set to everyone.
We should thank Kevin and Nyangiri as well.
Excellent. I didn't know you could do that. So that's great. That's super cool. And I'll also put this comment as well. Okay, so now you're interested in looking at the different cell lines in that data set. So you can use the function cellinfo, and you specify the pset name, so your object, which is CCLE. Oops, you just want, head of that. Okay, so it's still a little bit messy because we tend to keep all the information that was available in the initial study. But there are 2 columns that are mandatory in Pharmacogex. Is the cell ID, that's the official cell line name in all the Pharmacogex objects. So if we basically standardize the cell line name using the Celosaurus database, it's the biggest database of cell lines in the world. And so you can basically that's a way for us to be very standard when it comes to cell lines. So cell ID is mandatory, and tissue ID is also mandatory. And we use the Oncotree nomenclature from CBARPL to basically name the tissue types of those cell lines in a consistent manner across all the different data sets. You can see here an example. So for instance, if you're interested in, the tissue type distribution in that data set, you can look at, oops, sort that. So you can see that we have 192 cell lines from lung, 188 from hematopoietic and lymphoid tissue. So I'll cut and paste that in the chat window. So you can do your fancy pie chart or whatever you want to do to basically say, hey, there are a lot of lung cancer cell lines, a lot of hematopoietic and lymphoid tissue, quite a bit CNS, central nervous system cell lines, large intestine, breast, and so on and so forth. Some tissue type like salivary glands, small intestine, biliary tract, you can see that there are very, very few cell lines for those tissue types. So deriving cell lines is very difficult. It's not possible for all tissue types. But you get a pretty good representation in CCLE. You can see you have a lot of cell lines to do your research here. Okay, so now we've learned a little bit how to access cell line annotation. Now you can look at the drug annotation, and that is a bit more standardized. Not that it was easy, but we've probably done a better job for the drugs. So you can use the drug info function. So I'll just get and paste that in the chat. And so we try to collect as much information as possible about the drug. So again, there is one column that's mandatory in Pharmacogex, is the drug ID function. So this is the official drug name in that data set. Actually, it's a mistake here. You should not see the drug ID underscore. The official drug name should be that one. So it's basically you remove the drug ID underscore, will fix that object. But so if you look at all those drug names, those are going to be the same drug names across all the different pharmacosets, all the data sets in Pharmacogex. So again, very convenient if you want to have multiple data sets for the same drugs and so on. And so we've kept a few more information there. You have the compound generic name, but you also have the brand name. It's very perturbing, very, very hard in the field to follow, because like clinicians would use this name because that's what they know. It's like, hey, that's the brand name. So I'm gonna treat my patient with tenespamycin. But most researchers would know tenespamycin as 17-AG. So it could be a little bit confusing. So we have both. We also have the target of the drug. So that's a MEK inhibitor. That's a HR protein 90 inhibitor, for instance, EGFR, IGF-1R, inhibitor. So lapatinib is an EGFR year 2 inhibitor, this kind of stuff. We also have some kind of non-standard characterization of the mechanism of action. This is really like, if you ask different people, they will classify the drugs differently. You also have the class, whether it's a kinase inhibitor, whether it's a targeted therapy. You have the highest phase for that drug to be in where it has been used or tested. So that drug could be either discontinued or it has been tested in phase 3 trial, which is a very high bar. This drug is only being investigated in a preclinical setting, so it has never been used to treat patients, this kind of stuff. And then you have the organization, which company is actually manufacturing that drug? Is it Pfizer, BMS, Novartis, GlaxoSmithKline, and the like? So we have quite a fair bit of information about the drug, but the only thing that we managed to really keep super standard across all the dataset is the drug identifier that has to be, unified. But we also, it's not in that table, but we also have the SMILES identifier, which is a 2D representation of the chemical structure. We have the INCHI, we have PubChem identifiers. So we'll add them in the drug info in the future so people can actually have access to those. But we are gathering all that information for drugs. So you can basically connect to all those kind of established databases to gather even more information about the drugs. So you're not able to get the drug info. Did you do this? First, you have to load the data small object, and then you have to kind of rename it like this. And then you do this. Let me know if it works. Okay, now let's do something a little bit more complicated. Let's try to get, for instance, all the gene expression data, for CCLE. So you can use that function, summarize molecular profiles, and I'll explain to you what it does. You basically give the name of the object. So in that case, CCLE, and you have to specify what the summary statistic is, and I'll tell you what it is in a second. And hopefully, you have to define which molecular data you need. One, sick. Hopefully that works. Okay, smaller case, sorry. Okay, so what I've done here, I'll cut and paste that function first. Yeah, they should. So there is a comment saying that the download was took too long. Yeah, I told you to download the gray dataset, then I changed my mind. Sorry for that. But the point with the CCLE small is that you don't have to download anything. It's actually a small object included in the Pharmacogex package itself. So if you just load that object, data parenthesis CCLE small, there is no download involved. It's part of the object. So that's why I decided to go that road instead. So hopefully you can work on it now. So what does summarize molecular profiles? What this function is doing is actually it sounds simple, but it took a bit of work. It basically take all, let's say, the RNA sequencing process data that we have in that object. So that's why I had to specify which molecular data I wanted to look at, RNA-seq. Then we basically collapsed all the replicates using some kind of summary function. So we took the mean function. So we took the average of all those replicates. And then basically what we return is a very clean summarized experiment object where you have, I believe, let me see. So you have this clean matrix of 50 genes by 1061 cell lines. All the genes has been annotated with ensemble gene ID. And it contains the official cell line name that we have in Pharmacogex. So it just, if you're a machine learner and you don't really care about how to process the data, how to annotate those genes and so on and so forth, you just use that function and you really get that clean matrix without having to take care of all the creation, annotation, collapsing of the replicates. So it's just a very convenient function if you want to do your machine learning analysis. In the future, we'll improve that by actually returning a by using multi-assay experiment internally, so it's even easier to access those data. And you've seen that I've struggled a little bit to specify M data type. And that's because there is this internal naming of the different molecular data slots in the object itself. And so if you don't specify it, the function will actually issue an error listing all the different data types that you can access. I'm sure there is a function to actually display those. I don't remember top of my head right now. But the worst case scenario, you don't specify M data type, the function will tell you what are the different options, and then you just specify it. So in that case, I took the RNA-seq experiment, RNA-seq data. If you want the mutation data, you use exactly the same function, but instead of RNA-seq, you basically, oh, actually there is no mutation, sorry, in that example object. So CNV, if I want the copy number variation, I do exactly the same thing, but I specify that I want CNV instead. And those are log2 ratio copy number variation. So summary stat mean works in that case. And then you get all the CNV data. Okay, I'm almost done. I would like to show some drug dose response data, and then I'll pass on to Levi. Okay, so now we know how to look at the cell line information, the drug information. We know how to extract, let's say, gene expression or copy number variation data from this Pharmacoset. Let's take a look at the drug dose response curves now. Oops. Okay, so here, if you look at drug dose response, the function drug dose response curve in Pharmacogex, you can see that you can basically specify a Pharmacoset, a data set, or even a list of them. I'll try to show you an example. You have to specify a drug and a cell line. And if you do that, you should be able to display the drug dose response curves in that data set. So let's give it a try. So in terms of sets, we'll just provide the CCLE. We'll see if that works. In terms of drug, let's try lapatinib. Obviously, those should be the drug ID that you got from drug info. In terms of cell line, it should be the name of a cell line. So I hope that is a correct cell line identifier. We'll check if not. And I think for the moment, we'll just keep it as it and see what kind of plot do we get. Okay. That's interesting. Okay, that cell line has not been tested with that drug. Hopefully that one works. Okay. So here, I basically said, hey, give me the cell viability data for that drug and that cell line, sorry, MCF7 elapatinib. And I kind of knew that there would be data for MCF7 because it's a breast cancer cell line, so elapatinib is a drug used in breast cancer. So sometimes they only test drugs in their tissue, in their cancer types they're being used in the clinic. And so here what you can see is that you have actually, we extract all the cell viability data from the data set and plot it here. So x-axis, you have the increasing concentration. Y-axis, you have the viability. So as it should, it started at 100%, and then it slowly decreases. The IC50 is probably around more than 1 micromolar, which is not great. So that drug is not really killing the cells very fast. 10 micromolar is a very, very large concentration of the drug. So usually an IC50 more than 1 micromolar, we don't consider that drug to be very effective. Let me see if we can actually fit the curve. Okay, plot type, I'll fit it. Let's try this. So we also pre-computed, we fitted all those curves using usually 3-parameter logistic functions. You can actually design your own functions, and that would be the fit for that curve. So it's constrained to not go over 100%, so it's fairly flat, and then you see some inhibition at the very end. I don't remember how to display all the IC50 AUC, but I'll show you how to access those data. So you know what? I'll cut and paste that function in the chat window. Okay. So now, how do we access the actual IC50 AAC, like those summaries of the drug dose response curves? So I believe there is a summarized sensitivity profile, so very similar to what we had before. Actually, I'll look at the whole page. Okay, that's the case where it has been abstracted, so I use the double question mark. I select the Pharmacoset method. Okay, so you have to provide an object. You want basically, you have to say which kind of sensitivity measure you want, whether it's IC50, AAC, AUC. AAC and AUC is like 1 minus AAC, yeah, AUC is like 1 minus AAC. Which saline and which drug. And you also, if there are replicates, you also have to specify how you collapse those replicates, whether you take the average, the median, or just the first or the last, the max or the min. So let's try this. Specify the object. I specify the drug. And I will specify the saline as well. And I won't specify sensitivity measure. We'll see what it should comply and give me all the options. Oh, it doesn't. So it takes by default. Okay. So if you want the summary measure, oops, you have to choose. So let's say IC50 recomputed. So you have two kinds of, for each matrix, we sometimes have two options, recomputed, oops, and published. So published, we just grabbed, oh, it looks like we didn't include it. So published, we just grabbed whatever was published in with the paper. So we didn't try to recompute and refit the curve. We usually use our recomputed matrix because we are more confident that we know exactly how it has been computed and it was uniform across all the different data sets. I don't know why I have trouble to get the right summary measure. Let me see. Or maybe we just shrink that small object. Let's see, AUC. Let's try AUC publish, see whether we included it. Nope, okay. So it looks like we only have AUC recomputed in that data set, so apologies for that. If you download the full data set, you should have access to all the matrix. Okay, I give up. If I put the default, it's gonna grab something, I just don't know what it is, which is bad. Yeah, that's an AUC, probably. Okay, I need to figure that out. I'll cut and paste that function. There you go. But the bottom line is that we summarize sensitivity profiles. You should have access to all those summaries of drug dose response curves, whether it's IC50, AAC, And that's really what you use for a biomarker discovery. So I'll stop here. I'll update the vignette and share that back to Levi so he can share that with you. But hopefully I've showed you how to download, how to see what's available, how to download the data set, how to see what kind of cell lines and drugs are in the data set, how to plot the drug dose response curves. What was not very convincing, but how to get the summary of those drug dose response curves as IC50 AAC, and how to get the molecular data. So if you're interested, don't hesitate to reach out. We have other workshops and vignette for Pharmacogex and the other packages, and we're always very happy to help. And more importantly, if you play with the data and you actually find errors or inconsistencies, don't hesitate to open an issue on our GitHub page so that we can follow up and make the data and the package better. So thank you for your attention. If you have any questions, I'm here.
Thank you, Ben. All right, so I'm going to take over now, and I'll start. Let's see. Let me just share my screen. Okay, here we go. Okay, I'm going to go pretty quickly over a couple of slides here just because I want to save some time to show you some things interactively, and you have access to the slides here. Just to say that there are relationships between all of these packages that I'm talking about. There's a combination of databases from the NIH and NCI, the GDAC Firehose, and the data structures that Ben started talking about, Multi-Assay Experiment, and also cBioportal. And between these, you have access to a really wide range of cancer omics data. This is a sort of a schematic overview of what we think how these things relate to each other in terms of the level of integration that they provide between different omics data types, ease of use, and completeness. And there's necessarily a trade-off between those things. And these packages, the one that my lab has produced, curated TCGA data and cBioportal data, are very high on the end of integration and ease of use, but low on the end of completeness. And I'll mention TCGA BioLinks here for going a bit further down the completeness path, and Genomic Data Commons, as Sean was mentioning, for being high on the end of completeness, but low on the end of integration and ease of use. The only catch about this workshop, and I wonder if I present this, are you still able to see my screen if I do this, or does this turn into?
Yeah,
we're seeing slides now.
Okay, good. The packages themselves that I'm talking about today are I don't want to say easy to use. They do take training and some time to do, but they're fairly minimal interfaces. We've sort of tried to make them as minimal as possible so that you do a minimum of commands to tell the package which data set you want, and it just returns it to you. But in order to then go to the next step and analyze them, you really have to know about these core data structures. And so I'm kind of jumping ahead of a workshop that would teach you about these core data structures which would allow you to then take these data that you have accessed and do analyses with them. But just as a quick overview, the key data structures are summarized experiment and the related structures range summarized experiment, and then the derivative of those single cell experiments. And these are for data that have a matrix-like representation, which is usually features, variables in rows, such as genes and samples in columns, along with metadata, phenotype data, clinical data, and other related things. And these are really the most widely used data structures in Bioconductor today for especially for transcriptomics, but for a range of different things. Another data structure that is relevant for the packages that I'm showing you today is ragged experiment, which is for things like segmented copy numbers, somatic mutations, the kinds of things that would be represented in BCF files, things where you have sets of genomic ranges that can differ in different samples. And honestly, each of these things, I could easily spend an hour just taking you through and showing you about these and how to use them. They do make your life easier once you learn how to use them, but they make it a bit more difficult when you first start because they're each their own particular things. And finally, multi-assay experiment, which is an integrative representation for multi-omics data. And it is something for experiments that have several different types of data in them, such as the Cancer Genome Atlas, or anything where you have, for example, transcriptomics plus somatic mutations plus methylation, or some combination of experiments. And it's modeled sort of after summarized experiment, so that the things that you do to summarized experiment, we have tried as much as possible to make those also usable for ragged experiments and multi-assay experiments. So you learn as a set of actions, and they're shared as much as possible across all of these different data types. So just to say, you know, I'm going to go through some data packages fairly quickly today, but in order to really make use of them, I'll point you to another workshop on these as well. I think you would really have to do that as well. And just a little plug here, why use these data structures? They save a lot of work in data harmonization and management and make it less error prone. And we let people like Marcel Ramos do that sort of very complicated work so that we don't have to. And the first test case for multi-assay experiment was the Cancer Genome Atlas, under the assumption that if we were able to integrate all of these data in the Cancer Genome Atlas, we should be able to apply it to any other data set. And it has proven to be quite a flexible package. Maybe I'll skip past TCGA biolinks. Other than that, I will say that if you're interested in a component of the Cancer Genome Atlas that can be represented as a summarized experiment, so in this example, I want gene expression or other things like this, TCGA biolinks is great. It is then quite a flexible way to, and in some ways easier than the other things for getting a summarized experiment. But if you do want a multi-omics data set, that integration is up to you if you're using TCGA biolinks. Our package, curated TCGA data, works with one function for both for searching and for downloading data. So in this example here, well, we'll go through this a bit before, but it allows you to search which data sets are available, which data types are available for each data set. This is adrenocortical carcinoma. And then to download this, in this particular example, I'm getting reverse phase protein array and RNA-seq for the adrenocortical carcinoma data set. And this command just downloads this as a multi-assay experiment, so that these profiles, the RNA-seq and the protein profiles, are linked to the clinical data and to each other already. And hopefully we'll have a bit of time to go through this, but I'll just say this same function can be used to get all of the data types for one cancer, or even all of the data types across all the cancers, or one data type across all cancers. If I select one data type and all cancers, I get a pan-cancer data set in one command here. So it's very easy to use in terms of getting the data in a pre-integrated form. The second package, and a more recent one that we have put out, is called cBioportal Data, and it's an interface to cbioportal.org. And this is a really great way to go, because cBioportal is both an extremely broad resource. It has more than 260 multi-omic oncological studies of more than 20 primary sites, and it does include the Cancer Genome Atlas in it. So it's less complete than what is in curated TCGA data in terms of clinical data and the data types, but it might be good enough for you, in which case you can use this one package for TCGA and some hundreds of other data sets. One maybe warning I'll give to you about this is that not all of these 260 studies can actually be imported. There are, some of them have issues. We do have a vignette that says, as of the time of writing, which of these can actually be imported with cBioportal data, and which ones cannot. And if you want one that cannot be, then you need to file an issue with us, and we'll prioritize trying to get that one to import. Basically, we got, I don't know, 2/3 of them to import with some work, and then it starts becoming more individual. There are small issues that make it difficult to integrate some of these studies. There are two types. There are data packs that get an entire study, and an API that gets subsets of a study and makes it easy to do certain other kinds of things, like just getting a set of genes or just getting clinical data, things like this, whereas the data pack is an entire data set for one of these one of the cBioportal data sets. But both of these create multi-assay experiment objects. Just some examples here, and here's a link to a related workshop specifically on these two. And also, sorry, this also has some introduction to multi-assay experiment, ragged experiment, and summarized experiment. I'm not going to get into the microbiome data today in our examples, but I'll just mention, if you are interested in the microbiome, these are two packages also from my lab that there's a similar sort of philosophy behind them of making data download very simple. So there's some tools there for browsing which data sets are available. Then there's just a single command that you type and you get the data set on your desktop. There are some major changes undergoing in the next Bioconductor release next month. Currently, this produces a list of expression sets, just like Geoquery did, but after the next release, this will be producing summarized experiments and something called tree summarized experiments. And another package called HMP16S data, which provides all of the one type of microbiome profiling data from the Human Microbiome Project as summarized experiments. And it also helps you to attach the phenotype data, which are in a controlled access repository of the NCBI if you apply for access to those. Okay, so I'm going to move now into the materials. So actually I will start with this multi-assay workshop just to show you briefly about these data structures that we're going to get. The multi-assay experiment is for one with each of these rectangles represents some different dimensioned assay. So they don't necessarily have the same rows or the same columns, but they're all connected to one set of what's called the call data, which is where clinical data and phenotype data go. And then these assays themselves, they can be matrices, they can be other things. In these, they are ragged experiments, summarized experiments, and range summarized experiments. And I'll just note there's an API cheat sheet. This tells you all of the things that are available to you once you have this multi-assay experiment. This is also available on the multi-assay experiment landing page on Bioconductor. So let's see, let's go over to here. And I'm going to skip down to curated TCGA data. And I'll just show how this generally works. Actually, I can call curated TCGA data with no arguments, and it's going to give me an idea of what's available without actually downloading all of these data. These are the default arguments, which I spelled out explicitly here, but you don't have to. Taking a little bit of time, but that's okay. I'll let everyone catch up a bit, and I'm going to try to make sure during this time that I have my chat window working in case there are questions. Okay, I do have the chat window available. Yes, multi-assay experiments is a class. Thanks, Sean. Not sure why that is being so slow. While that is happening, maybe I'll answer a question that came up before about finding studies. Curated TCGA data obviously only is for working with the Cancer Genome Atlas. There's a lot more in CBio Portal, and it is possible to search for studies using our package and especially the API client. But to be honest, to search for things, this is where I would do it, from cbioportal.org, because they have a fantastic website. It is the premier source for publicly available oncology data. So if I were searching for something like, let's see, lung, I do this, it shows me all of the lung cancer studies that are available. Let's see, I don't know, what am I going to do next from here? Let's select a couple studies and let's explore them. And so it has brought together those two studies. It's showing me a summary of them. the types of molecular profiles that are available. This is really, a much easier way to get an overview of the studies than anything that you could do through our packages. And you can even go to clinical data and get an idea of what clinical data are available for these. So you can go to CBio portal and search for subtypes, search for whether there's survival status available. For, let's see, what else do we have here? For all kinds of clinical and pathological data. Also gene panels, this is saying that, so this is only the impact 410 gene panel. This is not a whole transcriptome assay here. So we could then go from this. I mean, you can do certain analyses on cbioportal.org, but it's certainly nothing like the range of analyses that you can do in Bioconductor. So once we found this data set that looks interesting to us, then what we can do is go to, take this study and go about getting it into Bioconductor. So let's see how we're doing here. Okay, this did finish. So that gave me a list of IDs, titles, sizes, classes. So you can see lots of ragged experiments, summarized experiments, range summarized experiments. This is what is going to be contained in the multi-assay experiments when I do a call to curated TCGA data with some more specificity. So I'll continue with this a little more. Now, when I call it with one of the disease codes, so I saw here, there's, this takes a little bit of getting used to, but this first bit is the disease code. I think I can also find that with, what is it, study. No, it's, these are the data types that are available. Okay. Here's the list of disease codes on the main help page for the package. So you can see here that ACC is adrenocortical carcinoma. And I often use this one for an example just because it's smaller. If we go to lung adenocarcinoma, this is a much larger data set, as is the lung squamous cell carcinoma. But if I wanted to, let's see, I don't know, let's try, since there was a question about lung cancer, I don't know, let's look at LUSC, can find out a little bit about that, curated. So this shows us these different, these are where the actual data sets are stored in Experiment Hub, and the titles of each of these. You don't have to worry about this unless you want an individual data set. So if I wanted to get some component of this on its own, I can get it from Experiment Hub. But the way I would usually do this is, okay, I'm going to pick something small. Just let's see. Let's take, oh, this is moderate. If you're working on Orchestra, it's going to be fast in any case. If you're If you're at home, some of these would be a bit larger. I'll ask for mutations. Well, that's a bracket experiment, a little more complicated. Let's get the RNA-seq gene. So that would be so disease code. Or maybe I'll do this, all right, do this for LUSC and assays equals, and I'll call this LUSC RNA-seq. And now this is going to go to Experiment Hub and look just at the lung squamous cell carcinoma data set and just RNA-seq by gene, and it's going to create, look at that, it's faster to do the download than it was to see the outputs. Oh, I have to do one more thing. There's an argument called dry run equals false, and that means actually download the data, not just show me what's available. And now this is really downloading from Experiment Hub, and it's going to produce a multi-assay experiment. It downloads a number of different things. It's getting clinical data, it's getting maps, and then it's actually harmonizing everything. It's dropping clinical data for which there are no assay or no RNA-seq data. It's dropping RNA-seq data for which there are no clinical data, except there aren't any like that. And then it's producing a multi-assay experiment. In this case, it only has one assay in it, the RNA-seq. If I were to do this again, adding, let's say, something else, I forget what another thing in there was, so I'm going to do, let's see, I'll copy that, I'll add it, do another dry run just to see what data types were available again. Get rid of this argument. And I see, okay, there's microRNA, another small RPPA array, there's another small one, so I'll do that. So now it's going to go, it won't re-download anything that was already downloaded. It's made a local cache of those things. Here, I'll copy this into the chat in case you want to try this yourself. Oh, I called it that, even though now it has the RNA-seq and reverse phase protein array. Now, if I wanted to see what clinical data are available for that, that is something called call data. That is, I'll put this in something else called, I'll show it first, call data. Now, the problem with the call data here is that it's a lot. This contains all of the clinical and pathological data available from TCGA for this data set. And if you're someone who has analyzed TCGA data, you might know that they have several levels of clinical and pathological data. There's like one level that is the simplified most important variables, that's level 3, and then they have like level 2 and level 1, which are more and more detail, including things like all of these individual mutations, then things like day of creation, month of creation, batch codes, all kinds of different things. And this has merged all of them into one gigantic data frame. It has like 2,000 columns in it. So it's a bit overwhelming to look at. But what I would do, if I really wanted to summarize, to look at those 2,000 columns, might be to do something like convert it to a data frame. So this is a capital D. I don't want to get into too many details here, but if you want to view this in RStudio's browser, it has to be converted to a data.frame, and then you can use view for it. I can also just do, for now, just something like 1 to 5, 1 to 5, and see the start of it. And you see these are, we have generally put the most important things, these level 3 things, right at the beginning, so you can, they're easier to find. And then at the very end, there are some subtypes which we've curated from the original papers. Since I think maybe that is all I will do for curated TCGA data right now. And then I'm just going to mention a little bit about CBio portal data. I've included a link here to an interactive workshop on CBio portal data, which Marcel Ramos made. It actually, I love it. Let me just show you. See if I. You can go through this, and it will ask you questions, let you type in code, and tell you when you have the right answer for your code. It uses a package called LearnR that's for doing interactive tutorials by yourself with CBio portal. So and it's running on Shiny, just to show you that. But to start with, I mentioned there were these 2 different ways of accessing data, the CBio data pack. These are the full data sets pre-packaged, and then CBio portal data, which uses API queries. So maybe I'll just, I won't use this. I'll let you use this. It's fairly safe, self-contained, but I'm going to look at $CBio portal data. Let's see, oh, I need to library CBio portal data first. So maybe I will, let's look back at here. Sorry, the CBio portal at this data set that we found that we were interested, lung MSK PDX. And I'll demonstrate 2 different ways of getting this study that we found that we're interested in. So the first way is the easier way, so I'll do that first, the CBio data pack. And I'll go ahead and add this here. So CBio data pack, and the cancer study ID is like this, and I'll call this MSK PDX. I'll just call it that, that's fine. Oh, remember how I mentioned that not all of the CBio portal data sets actually work? this is telling me that it's not actually building. So I can try this, but I'm probably going to get an error. I don't know. I don't want to try it. Well, I'll try it. I'll show you. It's not going to work. So it's downloading this data set with everything. And then, yes, it's saying could not find a seek names column. So there are a number of oddities in the CBioPortal data sets. In this one, there were things that looked like mutations, but we couldn't find a column automatically that looked like that looked like sequences. So let's try another one and see if we have better luck. This is the risk I take for doing things fully interactively and taking requests for data sets. Okay, here's another one. Let's see if we have any better luck with this one. I should add that if we really want that study, actually we can still access it through the API, and I'll do that. I'll do that in a minute. So it's all is not lost. We can still get this data set. So this is lung SMC 2016. Let's see if this one works. No, also not working. Okay. No, I'm not going to proceed. And yes, this is a nice thing as well. I can download the study anyways and also work on the components myself. So it is, if you really want a data set and don't want to wait for it to be supported, then this is what you would do. You download study, it will give you all of the data files that come with it, and then try to convert those into a multi-assay experiment or do your analyses on those files directly. If this were one of the working studies, I could go here and see one. Here's one that'll work. MA, because it's in the example, I know this one works. What's going to happen is it will download the study file. It will take all of these files, import them into R, create summarized experiments and ragged experiments, and merge those with the clinical data and create a multi-assay experiment. So there's a lot going on in this one command. This, honestly, if I were to download these files and then try to create a multi-assay experiment with it, it would take me some hours to do. It's often, there's a lot of integration. So we have 11 data sets from this that have been integrated into one multi-assay experiment. I can do things like row names, MAE. This shows me, that we have gene symbols on some of these. The copy number doesn't have row names. I can do call names, MAE. These are the columns from each of the assays. So, these are TCGA studies. I can also look at call data. So this has only 107 elements, so it's somewhat more, or columns, so it's somewhat more manageable than what we got from curated TCGA. It doesn't have all of those levels of data. It just has the most important things. Okay, let's take one of these and try to get it based on the API. So I'm going to go back here. This shouldn't take too long, and then I'll finish close to on time. Let's see, I'll just search CBioPortal data. Okay. So using the API, I need to do an API command or create an object that is from calling CBioPortal data. Let's see. Sorry, it's just CBioPortal. And just to show you, This command doesn't do anything except connect us to their service for getting data. And one thing I'll just notice about this, CBio Portal, it's a website, but it's also an open source software project and one that many cancer hospitals use for hosting their internal data as well. So I've set this up to connect to cbioportal.org. But if I were working at a hospital that had an internal installation of CBioPortal, I could also use this to access our internal data that we were hosting internally for for internal research. But once I've done that, that defines our connection, and now I can do things like get studies from this API. Equals API, okay. This shows us, it's actually 309 studies now. And one of these, I'll do view. One of these should be, what was this? Lung MSK PDX. Let's see if I can find this in here. So it's there. There's the study ID and just some other information, description, and things about this study. So now I could do this next line, clinical data. I'm just copying this again from this help page from CBio Portal. Clinical data, API equals API. I could have called this something else that might be less confusing, but. And there I've accessed that clinical data. This should be the same as what we saw on cbioportal.org, except that we have it in Bioconductor now. Let's see. I saw that that was a that was a gene panel. I'm just going to go back now to the help page. For here, and I'll take this as an example. Except I want this data set. What was the panel that I saw there that I liked? It was, oh, I'll go back to, I lost that. I lost that gene panel. Here, I will try this first. No, I need to. Oh yeah, just to say it's not 100% of data sets here, but it's 96.6% through the API. So it's a somewhat higher percentage through the API. Sorry, I'm going to stick with since I'm close to out of time, a working example. And show what it would produce. Sorry, I called this API. So for this particular data set, again, the adrenocortical carcinoma, it's accessed 3 different data types: protein, copy number alteration, and mutations, and created a multi-assay experiment from those for me. It's limited to this AmpliSeq panel ID, so we have only a small set of genes. If I look at row names, ACC, TCGA, this is clearly not the full data set. So we have TP53 and a few other genes, but it's a subset of the data set that we get through the API. You actually can't get the entire data set through the API. You have to limit it to some set of genes of interest. So I think I've gone over time. Are there any questions or requests for a last minute thing before we sign off? And I'll say that the question that was asked before about, I actually had never heard of an ER positive subtype of lung cancer before. I had to look that up afterwards and saw that it actually is a thing. I don't know if it would be annotated as such in the clinical data of any of these things. We might have to actually just look at the gene expression data sets to look for tumors that were expressing the ER gene, which I didn't do today. But if I were to do that, I would take the RNA-seq data set or download a multi-assay experiment that included the RNA-seq data set, maybe also the reverse phase protein array data set if we're talking about TCGA. If it were something else in cBioportal, I would try to get transcriptome data, identify the samples that had highest expression of RNA-seq, and then potentially subset the data set according to that. And I could probably do that using cBioportal data if it were in a data set that we're downloading correctly in a half an hour or something like that. You're very welcome, Elizabeth. But yes, cBioportal is fantastic, and it is where I would go to start looking for something like that, and using the clinical data, the summary, and things that they offer. So I want to thank all of you for the organizers for inviting us, and for all of you for attending this 3-hour workshop. Those of you who stuck it out to the very end, I really appreciate it.
Yeah, so yeah, then thank you to all of you for offering this training section, and to the participants as well. Right after this meeting, the recording will be made available, and we will also upload it on the H3Africa YouTube channel, and then we will share the link to everyone. So I try to keep track of all the various links with the course materials, and I've also populated it on the Discord platform. So yeah, you have them on the Discord platform as well. Once again, thank you. And the session continues tomorrow, same time. We'll have the epidemiology course for tomorrow. Great. So I think.
Thank you, Francis.
Thanks, Amy. Thanks all.
Take care, everyone. Keep in touch.
Yes.
Bye. All right. So we sign off.